So this is the Staph and Strep lab. And so I just want to go over basics and make sure that you understand it. I know you guys can read it, but I'll just walk you through it. Okay, so the first thing is that I need you to remember that Staph and Strep are both gram positive, and they're both gram positive cocci, right? So what shape and arrangement would Strep be? So I want you to think about that. Okay, so it would be streptococcus, right? So strepto is in chains and cocci are the round ones. Okay, and then staph will be clumps, right? Or clusters, and they're cocci. So it makes sense. Okay, now when we do this lab face to face, usually we do a throat and a nose swab, and that's not a good thing to do right now. So I'm just gonna talk you through this lab and what you would see. Okay, so to inoculate the plates, we're gonna take the cotton swab it's like a Q-tip, right? We're gonna take the cotton swab and that we swabbed your nose or your throat with, and we're gonna do an initial streak on the plate. So we just kind of go back and forth with it. Then you take your loop and you'll do a streak for isolation from that. Okay, and then we do a few stabs in the auger too. It just makes it easier to see some of the colonies. We want to get single colonies. So what we do is we incubate the plates upside down for 24 hours. Okay, we're going to have two different kinds of media that I'm going to talk about. So we have MSA, which is mannitol salt agar. So mannitol salt. And we'll have BAP, which is blood agar. It's sheep's blood agar. Okay, so here's the two types of media. Oops, I kind of cut off the bottom of it. Okay, so blood agar is one type that we're going to use, and it's sheep's blood. So it contains 5% sheep's blood, and it's put in tryptic soy agar, which is the normal agar that we usually use. Sheep's blood is good at the blood works well so that we can see what we're looking for, so we can see hemolysis well. Okay, this is a really rich media, so it has lots of nutrients in it. So it's good for fastidious organisms. So fastidious organisms are organisms that need extra nutrients to grow. They can be hard to grow without extra things. They're kind of like picky eaters. Okay, now mannitol salt agar is the other kind of agar that we'll talk about. And the carbon source for it, so the sugar is mannitol. Okay, so that's one of the sugars that we used when we did the biochemical tests. One of the things that's special about it, besides the mannitol, is that it has 7.5% salt. So it's got a very high salt concentration. It has phenol red in it, and you've heard of this before, so that's a pH indicator. So it's red when the pH is neutral, and it's yellow when the pH is acidic. Okay, so that means that if the organism can use mannitol, okay, it will turn yellow. So every time we ferment a sugar, it produces an acid. So if it can use the sugar, it's going to ferment it and it'll produce an acid. Okay, mannitol salt agar is selective and differential. So selective means that only certain organisms are going to grow on it, and differential means that we're going to be able to identify different organisms. They're going to look different. Okay, so mannitol salt agar, so we use it to isolate staph, and then we can differentiate staph with it. Okay, so this is kind of a repeat, right? So it contains the carbohydrate mannitol. So mannitol is the substrate. Okay, it has phenol red, so it's going to be yellow when it's acidic. Okay, it's red when the pH is neutral, and it will turn pink if it becomes basic. So 7.5% sodium chloride is what makes this media selective. So the salt concentration is high enough that only staphylococci will grow on it. Okay, so if you think about your skin, okay, staph grow on our skin. They've developed the ability to, or they have the ability to survive in high salt concentrations. So that high salt concentration will kill off everything. So that's what makes it selective. Okay, so if you take a plate, so this is an uninoculated plate and it's kind of red looking because of the phenol red, and you inoculate it with Staphylococcus aureus, you see this golden color. Okay, so Staphylococcus aureus is going to ferment the mannitol and it makes it yellow. Aureus, okay, is gold. Okay, so AU is the symbol for gold, and aureus is a gold coin. Okay, so this means you can kind of make the connection, right? It's golden or yellow. Okay, now we'll keep going. We'll go back. So blood agar is sometimes called sheep's blood agar or BAP or BAP. Okay, so it's 5% sheep's blood and tryptic soy agar. And how we're going to differentiate the bacterial colonies is based on whether they can break down red blood cells. And we call that hemolysis. Okay, so hemo is blood and lysis is breakdown. Okay, so hemolysis is a breakdown of red blood cells. Okay, blood agar is used because it's very rich and it's good for fastidious and non-fastidious organisms. So if you had to give me the name of an enriched media, you would say blood agar. Okay, now we can look at how streptococci grow on blood agar, and we can differentiate them by the hemolysis, by how they break down the blood, so what color we get. Okay, Staph aureus will also grow on these. So not only will strep grow on these, but staph will grow on these. Anything will grow on it. It's really rich. Everything likes to eat it. Okay, Staph aureus also breaks down blood, so you can get hemolysis patterns with it. So you have to make sure that make a slide and do a stain. Okay. There are 3 types of hemolysis. Okay, so some gram-positive cocci, so streptococci, produce exotoxins. Okay, so they're going to be excreted from the cell, and they're called hemolysins, okay, or hemolysins. So they can break down red blood cells and break down hemoglobin. Okay, so there's 3 types of hemolysis, 3 major types. So the first one is alpha. Okay, so this is alpha. And see, it looks like an alpha. Okay, and alpha looks green. Beta is this one, and it looks clear. And then gamma is no hemolysis. Okay, so 3 kinds of strep will produce these 3 patterns, these 3 hemolysis patterns. Okay, so alpha, beta, and gamma. So if you take a swab, you put it on the plate, you can look at the plate and you might see some colonies that look green around them. You might see some colonies that look clear around them. You might see some that aren't doing anything to the blood agar. Okay, so Streptococcus pneumonia, okay, does alpha hemolysis. So its colonies look kind of green. It's not a pretty green, it's kind of a dark green. Okay, so the green is due to the partial breakdown of the red blood cells and the hemoglobin. Okay, so you guys learn about that in anatomy when you talk about bile. Okay, so the colonies, if you look at them, they're mucoid, they're very sticky. Okay, so they have a capsule around them. So if you guys remember, here's the cell, and when some cells have a capsule, so it's a glycocalyx, so it's sugars and proteins that are stuck to the outside surface of them, it makes them really sticky, and it increases the pathogenicity, right? Because it can stick to your body, stick to parts of your body. Streptococcus pneumonia is also sensitive to this antibiotic called Optogen. Okay, if you make a slide of this, you're going to see gram-positive diplococci or short chains. Okay, this is a normal flora found in your respiratory tract, but it could also cause pneumonia, and it's referred to as pneumococcus. Okay, if you grow this and you see green, okay, you can then put an optogen disc on the plate, and if you get a zone of inhibition around it, that is another indication that it is the strep pneumonia. Okay, optogen is not a treatment, it is just a diagnostic. Okay, so here's another picture. So differentiation of streptococci. Okay, so identification of strep pneumonia can be made with optogen sensibility test. So we got this zone of inhibition around it. Okay, so it only affects this pneumococci. So it doesn't affect any other alpha hemolytic streptococci. So that's one way to know that you're dealing with strep pneumonia. Okay, so it's real sticky. Okay, now there are other alpha hemolytic streptococci, and they're in this group called viridins. So viridin is green. Okay, so it's a group of several species. It's very dry, it's not sticky looking. It's not sensitive to optogen. Okay, so it's not going to give you a zone of inhibition around it. So no zone, shoot, I'm trying to get a black. No zone of inhibition around it. It can cause cavities. It can cause endocarditis. Okay. Or they're a group. It's not just one. Okay, now beta hemolysis is the total breakdown of the blood. Okay, so you can see very clear zones around the colonies. Okay, so it's a complete destruction of the red blood cells. So it clears the media around the colonies. Okay, so in order to differentiate strep pyogenes from other streptococci and some enterococci, what we're going to do is you're going to test the isolates for resistance to bacitracin. Okay, so if a bacterial isolate is beta hemolytic and bacitracin and sensitive to bacitracin, it's presumed to be Streptococcus pyogenes. Okay, so you get the zone of inhibition. Okay, Strep pyogenes is in this group of they're called Lancefield groups. Okay, so Lancefield, and I think I have this yet. So Lancefield grouping was developed in the 1930s, and it was a way to organize and characterize these beta hemolytic streptococci. Okay, so if you do a swab, you have these beta things that do beta hemolysis. They could be a lot of different things. So what they started to do is they started organizing them. And the way that they organize them is based on antigens that are on their cell walls. Okay, there's a whole bunch of groups, but the important ones, okay, group A is Streptococcus pyogenes. Okay, that's the organism that causes strep throat. It's sensitive to bacitracin. So you're going to get a zone of inhibition around the bacitracin. Okay, group B, strep, is Streptococcus agalactae. Okay, the reason that this one is important is pregnant women, you get swabbed for this before you give birth. Okay, and if you have it, if you're group B strep positive, they will give you an antibiotic to get rid of the bacteria before you have the baby. Because if you have a vaginal birth, the baby could inhale it and it could cause pneumonia. Okay, if you have the baby before they swab and before you take the antibiotic, then they'll treat the baby with antibiotics to prophylactically to make sure that they don't get this pneumonia. Okay, there's also group C, group D. So, but the two big important ones that we're really concerned about are A and B, because one causes strep throat and one could cause neonatal pneumonia. Okay, so gamma hemolysis is no hemolysis. It's just growth on the plate. Okay, so you just have, if you did a gram stain of these, okay, you would see streptococci. Okay, but there's no hemolysis on the plate. Okay, there are also biochemical tests that can be used to characterize these organisms. Okay, so there's more than one way to get to the right answer. Here is a key that would give you to try to take you down the right path to figure out what organism you have. Okay, so the first thing you do when you do get a swab and do a plate is the first thing you're going to do is you're going to gram stain the colonies. Okay, if you have a gram-positive cocci, the next thing that you're going to do is a catalase test. And I'll show you catalase test in a second. Well, I can show you now. A catalase test is just adding hydrogen peroxide. So H2O2 is hydrogen peroxide. If a culture has catalase in it, the hydrogen peroxide is going to bubble. Okay, so this is a way that we can differentiate between staph and strep. So staph will bubble and strep will not. Another way to do it is to make a slide and look at it. Okay, so if it's catalase positive, okay, so this is peroxide. If it's catalase positive, it's staphylococcus. If it's catalase negative, it's streptococcus. Okay, so if it's staph, and you can confirm that with your gram stain, you will do a coagulase test, and I'll show you, let me show you what that is. Okay, so there's more about catalase. Okay, the coagulase test. So coagulase is an enzyme that Staph aureus produces, and what it does is it takes fibrinogen and it turns it to fibrin. Okay, so fibrinogen is found in blood plasma. It can dissolve, right? So it's soluble. It gets converted to fibrin when we need to make a blood clot. So we have a way, okay, to add fibrinogen to the colony. Okay, well, here, let me show you. We can add fibrinogen, okay, to the colony in some water, and we're going to look for clumping, just like when you do blood typing. Okay, so if it's positive, it's going to clump. Okay, and clumping is also referred to as agglutination. That's another word for clumping. Okay, so if you have coagulase, you will have clumping. Okay, so if we go over here, go back to this chart, so you test for coagulase, you're looking for clumping. If it's positive, it's Staph aureus. If it's negative, it's probably Staph epidermidis. There are other things that you're going to see, right? So if it's on mannitol salt, it will have a gold pigment. It will also hemolyze or break down the blood if it's on blood agar. Okay, Staph epidermidis, it'll grow on mannitol salt, but it's going to look white, and it doesn't break down the blood in blood agar. Okay, now for the strep species, the first thing that we're going to do is we're going to look at how their hemolysis pattern. Okay, so alpha is green, beta is clear, and gamma is no breakdown. So there's no blood breakdown. Okay, so if you have something green and you want to see if you have strep pneumonia or one of these viridans, what you do is you add an optogen disc to it. If it's sensitive to the optogen disc, okay, then it's positive. If it's not sensitive, it's negative. Okay, so then if you have beta colony, you're going to add bacitracin disc. Okay, if it's positive, so that means it's sensitive. It's Strep pyogenes. So this is the organism that causes strep throat. Okay, and then if it's negative, it could be any of the other streptococci. Okay, so group B, group C, group D, group G. This one is group A. Okay, and the gamma, the example that we use for gamma is Streptococcus faecalis. Okay, now this just walks you through the tests. Okay, so there's an example. So what we do is if you take peroxide and you add it to catalase, we get water and we get oxygen gas, so we get bubbles. Okay, coagulase test, if we get clumping, it's positive. Okay, there are other flora that we're going to find in the nose and throat. So if we were doing this for real, you could possibly see some Neisseria meningitidis. Those are gram-negative diplococci. Okay, so these are the cells. These are the bacteria. Okay, and they are actually oxidase positive, which we didn't do the oxidase test. Okay, now also in your nose and throat, you have these things called diphtheroides. So diphtheroides are in this group called Corynebacter, and they're normal flora. Okay, they don't produce any toxins. They don't cause any illness. They're not a big deal. They're just in there. Okay, they're gram-positive. They're pleomorphic. So pleomorphic means that the rods can be different sizes. They don't move. They don't form spores. Okay, and that's that.